I want to jump to a couple of the maybe one or two audience questions at this point, but I want to have Alok or Russell. Are there one or two here that speak to you in terms of what you think you'd be able to speak to? We've got a few on stability. We've got a few on the IP landscape, which is pretty hot. Are there any that you're most passionate about digging into?
I can take the
questions from the Q&A.
From the audience, yeah. Alok, go ahead. What one were you looking at?
Yeah, I can take the stability one, right? The question 5, I believe.
Does formulation help in stability? Yep, of these LNPs.
And I think, yes, it's critical, right? The composition is everything when it comes to the LNP product. It typically is a mixture of 4 lipids in different ratios, right? And your product has to be stable. And remember, these are hydrophobic in nature, all 4 lipids, and how you balance it also affects the potency. For instance, right, PEG. I mean, I can put all the PEG I want, but at some point the potency falls off a cliff, right? So you can't use all the PEG in the world to stabilize an LNP. So it's a very delicate dance between the 4 lipids that you have to play. And I would say it's better to get an early read on how stress your formulation and see where the breaking points are, right? Because it's not good to reinvent the wheel once you're deep down in the preclinical, you did all the mouse and the rat and the monkey studies, and then suddenly you say, okay, well, this is not stable. And then you change it, the whole property and biodistribution will change. So I believe, formulation is the key in everything that you do as an LNP.
So can I add to that a little bit? So so stability is I mean, and kind of the build up to this, I kind of wrote down kind of a lot of notes, and actually stability kept coming up, right? And there's a couple of reasons. One is you need kind of immediate stability to reach your target tissues. And then you need product stability, right? And both of those are actually fairly important. From a vaccine standpoint, if you want the broadest impact of your vaccines, you need product stability, right? And you need to understand where that's coming from. I think there's a few arguments there of where product stability originates from. I think that it's very clear from publications that mRNA design and the vehicle composition and the types of cationic lipids you're using has, and also excipients that you may add in, has a profound effect on the mRNA stability. But if you go back to Onpattro, actually, it's a pretty robust LNP. Actually, if you just use that base formulation, it's room stable for a few months, really. But the payload would not be, and that I think is something that's really interesting. And you do see some changes in the LNP, right? Maybe some Oswald ripening or something where you see a little bit of change in size or dispersity of size. But the reality is, for the most part, that's staying pretty much, okay. But the payload itself is a critical kind of thing that you're always dealing with and wrestling with. And there's a lot of things that will influence that stability, including its design and then the environment of the LNP. So it's a great topic, and for vaccines, it's really, in terms of key questions as we move forward. And ways that mRNA technologies become impactful in the very long term. It's showing that you can have a very stable product in a very simple formulation.